Hello everyone. I'm really excited to be here today and really honored to be able to present some of the progress that we have made at Cincinnati Children's in engineering next generation organoid models. And also I will describe some of the efforts that we recently started to really transform organoid from basic biology tools to applications which are amendable to industry standards. But before I'm going to dive into more details, I would like to introduce you to the Center for Stem Cell and Organoid Medicine that I represent today. This center was established in 2017 at Cincinnati Children's to bring together world-class experts in stem cell technology. That includes development biologists, bioengineers, as well as medical doctors who now united can work together toward one common goal, which is acceleration of cures for major diseases of our times using stem cells and organoid technology. Currently, there is around 34 different labs under the umbrella of Custom, all of which are working on the induced pluripotent stem cells. So as you all know, induced pluripotent stem cells can be generated from the blood cells of basically any individual, either healthy or diseased patient. And thanks to the methods, many of which have been established at Custom based on the embryonic studies of embryonic, human embryonic development, now we can create from those cells three-dimensional miniature versions of human organs in the dish, which you know very well under the name of organoids. And we at the Custom were able to establish the methods to derive various parts of human gastrointestinal tract, including esophagus, stomach, small and large intestine, as well as hepatobiliary tissue, liver. But we also more recently started to work on organoids of human brain, lung, heart, kidney, and pancreas. These organoid models have shown to be able to really recapitulate the complex architecture as well as biology of native human tissues in vitro, and therefore we can use them to study human development and disease. And at Custom, we also use them to create organoid-based platforms for drug discovery and development, so safety, efficacy assessment, the preclinical stage of development of drugs, as well as more recently we started to qualify the use of various types of organoids, including intestine and liver, for transplantation-based approaches for the treatment of end-stage disorders. So for example, we are qualifying the use of intestinal organoid for the potential compassionate use in patients who refuse transplant, right? So transplant rejection. But today talk, I will focus just on a subsection of the research that we are doing that aim toward development of next generation of organoid systems with increased complexity and functionality. So we have developed a various of methods to be able to incorporate multiple different tissue types into one organotypic model, what helps us to really emulate organ level functions such as, for example, peristaltis. And those methods can be broadly categorized into two groups, co-development methods in which we are able to derive from one pool of iPS cells multiple cell lineages that results in organoids that possess multiple different tissue types. So for example, epithelium, mesenchyme, and immune cells, and good example of that are human colonic organoids that we are able to co-derive with resident immune cells. We also use co-culture methods, so the second type of tissue engineering approach, where we firstly separately derive various cell lineages, precursors of various tissues from induced pluripotent stem cells in separate dishes, and then recombine them in vitro into one organotypic model. And in this way we are able to, for example, create vascularized or innervated organoids or organoid immune cell co-cultures. We also started to to develop methods to establish interconnectivity between different organoid models, as that would help us to really study interorgan crosstalk, that it's important, for example, to better understand more complex human disorders, such as, for example, metabolic diseases. And good representation of that, it's the world's first fully integrated multi-organoid system that was developed in Dr. Takeba's lab, where we connected human, or co-derived actually, human liver, pancreas, and biliary ducts in one system. Additionally, more recently, as I joined from myself pretty recently, this team around a couple of years ago from Emulate, where I also work on the organs on chips, we started to connect and really merge iPSC-derived organoids with this type of organ-on-chip platforms to additionally incorporate the mechanical forces, such as, for example, fluid flow, what enables us to study more dynamic processes occurring in the human body, such as immune cell recruitment. But the next few slides will focus just on showcasing 3 examples of next generation models that we are able to create. And the first one comes from the lab of Dr. Jim Wells, where we are able to create innervated intestinal organoids. And we have done that via the co-culture method. We firstly derived from induced pluripotent stem cells neural crest cells that then we recombine with maturing intestinal organoid that was grown in a separate dish, and that enables us to really recreate the neuroglial structure like in vivo human tissue that possess neuronal activity that you can see here as represented by calcium imaging. And most importantly, we were able to actually recapitulate the electromechanical coupling of various tissues, including this neural tissue as well as smooth muscle cells, would enable us to really emulate peristaltis, what you can see resulting here, contracting intestinal organoid in the bottom left corner of the slide. We applied the system to study the cellular molecular basis of Hirschsprung disease. This is a congenital genetic disorder that is characterized by this motility of the lower part of the gastrointestinal tract, and we are currently using this system to really seek for the potential therapeutic approaches for this disease in collaboration with various pharma partners. Another example that I wanted to share with you today, it's an example of where we use co-development methods. So again, just to repeat, from one iPSC pool of cells, we are able to co-derive multiple different tissue types. In this case, we are able to really co-develop human colonic organoids together with resident immune cells. And here you can see resident macrophages labeled in green that are coming out from this colonic organoids that show to be able to persist for a prolonged time in close association with colonic epithelium. And when we transplant it into animal, it's actually persists for a prolonged time. It's not being replaced by the bone marrow-derived monocytes. And these macrophages are capable of phagocytosing bacteria, which you can see here as appearing white signal, as we use pH-sensitive fluorescent probe to perform this imaging. And those macrophages are able to mount pro-inflammatory response to the LPS, which you can see here. They are also able to distinguish between the commensal and pathogenic bacteria, what I'm not showing here today. But all of those features really are characteristic of tissue-resident macrophages. And that really, this study laid the foundation for the application of this system into the studies of gut inflammation, and we are currently using it to really study and interrogate the mechanism of early onset IBD. Another way in which we can create organoid immune cell co-culture, it's really by co-culturing iPSC-derived organoids with peripheral blood cells derived from the same individual. And that's what we have done in the case of the human liver organoids. We co-cultured them with the PBMCs isolated from the blood of the same patient. And as you can see in here, up to 7 days of this co-culture, we could observe no differences in the morphology of the tissue. We also analyzed whether it's remained functional, and I can tell you that it does. Interestingly, when we compare this system to the allogeneic model in which we combine the iPSC-derived liver organoids and blood or PBMCs isolated from the blood of a different donor, leading to HLA mismatch in our system, we couldn't observe major differences in the morphology of it, right? So there was no significant death happening or toxicity, which was interesting, I think. But when we look closer into the cytokine and chemokine profile of those systems, we realized that in allogeneic conditions, we really do observe a significant increase in the release of T-cell effectors, so interferon gamma, granzyme B, as well as chemokine CXCL9, CXCL10, which are known to promote hepatic inflammation and migration of the leukocytes to the liver parenchyma in the context of liver injury or chronic liver disease, would suggest that we are really able to recapitulate the allo response in our in vitro system. And we are currently using this system in collaboration with Roche, a pharmaceutical company, to improve identification and characterization of subpopulation of patients at risk to develop immune-driven induced liver injury. So we are really creating the co-culture systems of human liver organoids and PBMCs from the donors that are carriers of the specific risk factors. So for example, specific HLA type that was previously also associated with the adverse drug reactions in the clinic, and we are trying to recapitulate it in vitro. It's too early to share the data, but I'm truly hopeful that this in the future could help us to avoid, this adverse drug effects to be happening at the level of clinical drug development. So in a number of publications, some of which are listed in this slide, we have demonstrated that the organoids can be used for modeling of complex human disorders, such as, for example, non-alcoholic steatohepatitis using human liver organoids. We can use them to perform personalized assessment of drug safety and efficacy, and also as tissue replacement therapies for the end-stage disorders. However, although organoids hold a great promise to transform drug development, precision and regenerative medicine, there is still a number of major challenges, and those are really listed in this slide that we need to overcome those in order to fully realize the potential of organoid research, and that includes a high variability in organoid culture. So they are variable in size, they are variable in numbers in between different wells. Their location, the hydrogel, also it's making development of downstream assays quite cumbersome. And they also are characterized, the workflows that we are using to generate and use this type of platforms are very complex. So it requires really many different experimental steps to be able to generate those. You need experts, users, and that of course results in a raw reproducibility of protocols in between different laboratories. Therefore, in 2019, at the end of 2019, we have established Custom Accelerator Lab that I manage, that it's really focusing on development of methods to improve scalability, reproducibility, in situ monitoring and characterization of organoid cultures, and all of that to really improve and accelerate the translation of organoids from bench to the bedside. So the first challenge that we decided to take on is to really transition our organoid models from this, small, low throughput dishes to large scale, high throughput and high content organoid screening applications, right? So we decided to implement automation of workflows. Here, what I'm showing you today, it's a fully automated, integrated system that we have built with the help of BioZero and molecular devices that it's quite sophisticated as it combines 9 different instruments, including incubators, liquid handlers, plate washers, readers, as well as high content imagers, which are all connected via the use of robotic arm and automation scheduling software. Each of these elements or instruments is able to perform one step of the organoid workflow, right? So we are now able to automate all of the workflow, starting from the cell seeding all the way to the downstream analysis of those cultures. But of course, that solves only some part of our challenges, right, as we are still using the matrigel, what makes development of assays quite challenging, as you know, that leads to the differences in the size, numbers of organoids across different wells, and makes just really assessment by imaging challenging. Therefore, we decided to couple it with newly engineered plates, which are called Grids 3D, and those plates have been originally developed at the lab of Matthias Ludlow, and now they are being commercialized by a small Swiss company, Sun Biosensors. And what is particular feature of those plates is that in every well of 96 well plate, you have a hydrogel-based microcavities. What enables us to really guide the aggregation of organoids from the suspension of single cells to create very homogeneous type of cultures of organoids, which are of the similar size and similar numbers across various wells, and they have very uniform distribution in 2D plane as well as Z-axis, which of course helps us to automate it. Additionally, they have the media exchange board, so really SLA format, fully capable to be coupled with any type of automation. And you can also take advantage of these plates in order to create the gel-free organoid immune cell co-cultures. So the previous model that I've been showing you, we are now transitioning to this type of setup. And thanks to those tools and methods, we are now able to perform a fully automated generation, maintenance, monitoring, drug exposure, as well as high content analysis of our cultures, in which what can be used to study disease phenotypes as well as compounds effect. And we applied these methods initially to human liver organoids, and here you can see the results that we are obtaining by its use. So as you can see, we can assess the drug effects at the level of basically single organoids thanks to the use of high content imaging and 3D image analysis, and we can combine it with the multiplex assessment of soluble factors which are indicative of liver health or injuries, such as, for example, production of albumin, release of LDH or AST activity. And with my last slide, I would like to acknowledge all of the custom researchers and PIs whose progress I was able to show you today. And I would like to say that we are very open for collaboration with both pharma as well as academic groups that are interested in really working together to further advance this organoid research. And we are currently a number of openings for talented scientists, including biologists as well as engineers. So if any one of you here in the audience today is interested, please don't hesitate to reach out. We really are searching for the people who could help us to translate organoid research from bench to the bedside. Thank you very much for your attention. I'm going to be happy to take any questions.